Good evening, students. We were learning about the photosynthesis. We learned about the processes. Then again we have to learn about the adaptations in leaf to perform photosynthesis. How the leaf is adapted. The first one is large surface area. You know that leaf is adapted to absorb maximum sunlight. If the surface area is large only, then the leaf can absorb enough sunlight. The second point, leaf arrangement. You have learned in botany or in plant life, you learned about how the leaf is arranged to absorb to get enough sunlight at right angle to each other, alternate or opposite arrangement of leaf you learned to obtain enough sunlight. Then again point 3 is cuticle and upper epidermis are transparent. You learned the structure of the leaf. Its epidermis, upper and lower epidermis are transparent. Then again numerous stomata to absorb enough carbon dioxide or to send out oxygen. Leaf is adapted. Its lower side has a lot of stomata. Then the thinness of the leaf is very thin, so it will cause the rapid transportation. Exchange of gases will be easier for the leaf. Then again the chloroplast. We know that in the upper mesophyll cell, in the upper cell, the palisade cells have more chloroplast inside it. Then the leaf has extensive vein system. Numerous veins are there. So transportation of water or minerals or the food made by the leaf is all transported very easily. It has a good vein system. These are the adaptations of leaf. Then let us see the end result of products of photosynthesis. What will happen to the products of photosynthesis? You know that the three things are produced by the photosynthesis, that are glucose, water and oxygen. Glucose, what happened to the glucose? That may be immediately consumed by the plant. Sometimes plant may use that glucose. Sometimes that may be stored in the form of insoluble starch. You know that starch is an insoluble substance. If we try to squeeze some rice or the boiled tapioca, we cannot dissolve it. Usually the starch is a insoluble substance, so it is stored in the root as tapioca or in potato, etc. It is stored as insoluble starch. Sometimes that may be converted into sucrose. You know that sucrose is the cane sugar. കരിമ്പിൻ്റെ പഞ്ചസാരയായിട്ട് സൂക്ഷിച്ചു വയ്ക്കാം. Or in other fruits also. We know that the sweet mango or other fruits also may contain or the beetroot, etc. The sweet sucrose that may be converted. Usually the cane sugar that is storing it as sucrose. Then it is used, the glucose, by adding some other substances or by polymerization. It may change into fat or protein. That is what happened to the glucose produced by the plant. Then again the water. Then water may be reutilized. You can see the word reutilized and may continue in the process of photosynthesis. That is happened to the water. Then again the oxygen. Oxygen may be diffused out. That is what happens. Some of the oxygen, if it is in the daytime, that is used by the plant for its breathing. Some of the oxygen is used by the plant. At the time of photosynthesis, plant need not to absorb oxygen from outside. Then again during the night time, usually the insoluble starch that may be stored as starch itself inside the leaf temporarily. But during the night time, that may be, you see, at night the starch is reconverted into soluble sugars, and that may be transported into different regions of the plant for its storage. That is translocated. That process is known as transported, meaning translocated. It is taken from the leaf and may be transported to the area of storage. Then again, factors affecting photosynthesis. Certain external factors are there, that are the light intensity, then carbon dioxide concentration, temperature, and water content. That are the external factors that are affecting the photosynthesis. You know that certain graphs are given here about the light intensity. You can see on one side, on one axis it is rate of photosynthesis is given, and the other axis it is the increasing light intensity also we can see. Increasing light intensity also we can see on one side. With the increase in water light intensity, you can see that at a certain level you can mark it as synthetic one phase is there. In that phase S1 you can see in that phase the when carbon dioxide concentration rise up to 0.02%, then that becomes stabilized. Then further increase in photosynthesis will not, rate of photosynthesis becomes stabilized after the increase in the level of light intensity. At a certain level, that rate of photosynthesis will become stabilized. Light is necessary for photosynthesis, but if we increase it to after a certain level, we cannot increase the rate of photosynthesis. That is what happened. Then again you can see here on one side another graph B, increase in light intensity on one side, rate of photosynthesis also you can see. You so in the previous graph 0.02 percentage of carbon dioxide concentration. Then again here we are increasing, or here it is increase the carbon dioxide concentration up to 0.05 percentage. At that light intensity increased and intensity of or carbon dioxide concentration is increased from 0.02 to 0.05 percentage. After reaching 0.05 percentage of concentration of carbon dioxide, again we can see the photosynthesis becoming stabilized. We cannot increase after a certain level, we cannot increase the rate of photosynthesis. Even though we give more carbon dioxide or more light, we cannot increase according to that. Increase in the amount of carbon dioxide and increase in the light intensity, we cannot increase the rate of photosynthesis. After a certain level, that will become stabilized. Then again, what is the role of temperature? Temperature, according to the rise in temperature, photosynthesis, the rate of photosynthesis also rise. But you can see its optimum temperature or most suitable temperature for the rate of photosynthesis is 35 degrees Celsius. That is the optimum temperature. That may be asked for the examination. Then for after giving this graph also several questions may be asked. You should learn what is explained in the graph. You should learn, understand about the graph. There may be, after giving this graph, the picture, after giving this picture, several questions may be asked. What is explained in the graph may be asked in the examination. Then the optimum temperature for the photosynthesis is 35 degree Celsius. Then again another fact is given here. Photosynthesis occur best at this 35 degree Celsius. Then again we can see after which the rate falls and stop above 40 degree Celsius. If we increase the temperature after the 40 degree Celsius, photosynthesis stops. That is happening to the how the temperature is affecting the photosynthesis. Then again here certain a rise of 10 degrees Celsius up to the optimum temperature 35 degrees Celsius double the rate of photosynthesis. For example, a rise from 20 degrees Celsius to 30 degrees Celsius or 22 to 32 degrees Celsius or 25 to 35 degrees Celsius double the rate of photosynthesis. Up to 35 degree Celsius according to the increase in temperature, rate of photosynthesis also increases. But after reaching the 40 degree, photosynthesis stop. That is happening to the rate of photosynthesis. Then again, you know that water content is also affecting the rate of photosynthesis. You know that if the water is not enough, the photosynthesis cannot done by the plants. Only 1% of water absorbed by the root is utilized in the photosynthesis. If the water level reduced much, the plant cannot do the photosynthesis. Then internal factors are also there. Chlorophyll must be enough for doing the photosynthesis. That is one of the internal factor affecting the photosynthesis. You know that chlorophyll need several formation. Yesterday in our last class we learned what are the minerals required for the formation of chlorophyll. Magnesium, nitrogen, carbon, hydrogen, etc. are necessary for the formation of chlorophyll. So if the plant has any nutritional deficiency, the chlorophyll will not be formed inside the plant body. That can affect the rate of photosynthesis. Then protoplasm also. Protoplasm, you know that the content of the cell. If the dehydration happen to the protoplasm, or the another thing is accumulation of carbohydrate. If there is enough starch inside the plant body or inside the cell, further photosynthesis will not take place. That is also another fact, internal factor. The amount of chlorophyll or amount of starch inside the cell or the amount of water inside the cell are the factors, internal factors that are affecting the photosynthesis. Then here another fact is given here. In some desert plant, they can do the photosynthesis even the night. That is one of the interesting things. Such plants are here aloe vera, tulasi, neem or people. They can give out oxygen even the night. That is another important fact. You might have indoor plants. About the indoor plants you might have heard. These plants can give out oxygen even during the night. Then again we have to learn about several experiments on photosynthesis. The test for the starch, that is usually learned in the 4th standard, how we can test the starch. Only a few steps are there for this experiment. First one is dip the leaf in boiling water. You can see in the beaker, you can first we can dip the leaf in a boiling water to kill the cell. So why, certain questions may be there after showing the experiment. Why the leaf is boiled in plain water? To kill the leaf. Answer is to kill the leaf. That must be right as to kill the leaves. That is the key words. Kill. That word must be there when you answer. Destroy the cells is not enough. To kill the cell is necessary for answering it. Then boil the leaf in methylated spirit. You can see methylated spirit, that is shown in the picture. In a test tube, methylated spirit is there. When we boil the spirit, we submerge that test tube in a beaker, then only we boil. that process is double boiling. That process is known as we are not putting that test tube directly. We should not put that test tube directly into the flame. Only double boiling is done when um, when we boil it in the methylated spirit. That is to remove the chlorophyll. Why the leaf is boiled in methylated spirit? The answer is for to remove the chlorophyll. Why the leaf is boiled in plain water? To kill the cell. Then again place it again in hot water to soften it. Then spread the leaf in a dish and pour some iodine solution. Iodine is brown in color, like the honey color. Iodine has the color of the honey. Then pour some iodine solution. Then the leaf will, if the leaf contains starch inside it, the leaf turn its color into blue-black color. What is the color of the leaf if it contains the starch? Such a question, you should write it bluish. You cannot write it as bluish color or black color. You should write it as blue-black is the color. The leaf turn blue-black. You cannot bluish, such if you write the word bluish, the answer you will not get the mark for that answer. Blue-black is the color you should write for the starch test. Then another procedure, for every experiment in the photosynthesis, one process is done. That is the destarching or removal of starch. For destarching the leaf, a plan for experiment on photosynthesis should be placed in dark for 24 to 48 hours to destarch the leaf. Destarch meaning removing the starch already present in the leaf. Then we will give a special condition for the leaf in the experimental setup. What is meant by the destarching of the leaf? The answer is there. A destarched plant is one whose leaves are free from starch. How we destarch a plant? By placing the leaf in dark room for 24 to 48 hours. That is how we are destarching a plant. Then the first experiment you can see to show that chlorophyll is necessary for the photosynthesis. Here we take a variegated leaf. Variegated meaning, here that word is there. Variegated leaf meaning the leaf having some green and some non-green part. In one of the board exam there was a question, what is meant by the variegated leaf? The leaves having some green and non-green areas. Then another question was also for the board exam. Write an example for variegated leaf. Here the examples of variegated leaves are there: collis, geranium, croton plant. Here you can see a variegated leaf here. Then destarch the leaf by keeping the plant in a dark room for a few hours. You know what is destarching. Then how after destarching, place the, you see the second point, place the plant in the sun. After keeping it in the dark room, then the whole starch inside the leaf is removed. Then we place the plant in the sun. After a few hours, pluck one leaf, make its outline on paper. You can see the design by placing an butter paper over it, and we can trace that design and keep it aside. Trace the same design and we can keep it aside. Then place the plant in the sun. After a few hours, pluck one leaf, make its outline on a paper and mark the green and non-green areas on the outline. Test the leaf for starch. You know how the starch test is doing. Here we saw how the starch test. First boil the leaf, then boil it in the methylated spirit. Then after removing that methylated spirit, again boil it in plain water to soften it. Then put a few drops of iodine on it. Then after that we can, when we put iodine, we can see the green colored part will change into blue black color. The other part, which color is shown by the non-green parts, that is brown color. This color, you must notice how these colors is brown. One is brown and one is blue black. The question may be asked, what is the color of the non-green part, what is the color of the green part. Such a question may be there after giving the experiment or the picture. After showing this picture, what is the experiment shown by the diagram, then which leaf is used in the experiment, then which is the solution to test the starch test, iodine solution, then the part containing starch, which color is shown by the green part, that is blue black color, and non-green part to show brown color. Such questions may be asked for the examination. After showing this diagram, you must able to identify each diagram. In the second experiment, you can see one destarched plant again taken, and a part of the leaf is covered by a black paper. Cover one of its leaf with black paper with a design cut is on it. Then place the plant in the sun. After a few hours, test the leaf which is covered by black paper for the presence of starch. You can see that the part which didn't get the sunlight will remain brown in color. The part that got the sunlight, that will change into blue black color after doing the starch test. In the second experiment, the experiment to show that sunlight is necessary for photosynthesis. On seeing the diagram, you must be able to identify the experiment, which is in the second experiment, question may be there, which is proved by this experiment. What factor is proved by this experiment, or what is the aim of the experiment? Such a question may be there for the second experiment or first experiment. You must be able to identify the diagram and answer the questions. Now I am finishing the lesson.